Module E, Group Sequential Trials with Multiple Objectives. I will begin with a longer introduction to go over the main themes that we will be exploring in modules E and G. As I explained in the introductory module, in addition to traditional, if you will, sources of multiplicity in phase 3 trials, and by traditional I mean multiplicity induced by the evaluation of multiple endpoints or multiple doses. So in addition to those traditional multiplicity components, it is increasingly more common to analyze multiple objectives in clinical trials that also employ data monitoring strategies at interim looks. Data-driven decision rules are applied at each interim look, and the clinical trial may be stopped as soon as enough information is accumulated to support a conclusion that the experimental treatment is superior to the control or may be inferior to the control. The fact that multiple endpoints are evaluated at 2 or more decision points, by which I mean interim and final looks, this fact contributes another source of multiplicity that of course needs to be accounted for. So in modules E and F, we will focus on the methods that are utilized in this setting to control the overall type 1 error rate across the traditional sources of multiplicity and also across several decision points. It will be worth mentioning that modules E and F mirror modules C and D, where we examined gatekeeping strategies used in clinical trials with parallel and general gatekeepers. The analogy here is that in module C, we started with a more straightforward class of multiplicity problems with several families of hypotheses. Specifically, we focused on problems with parallel gatekeepers. Module C provided a foundation for the discussion of more complex problems with general gatekeepers in Module D. And similarly here, in this module, we will begin with a more straightforward class of adaptive designs. We will first examine group-sequential designs, and with this foundation in place, we will be ready to handle multiplicity issues that arise in general adaptive designs, and we will do that in Module F. So back to the outline of Module E. As you can see on the slide, there will be 2 sections in this module. First, Section E, Introduction to Group-sequential Trials. In this section, we will assume that the only multiplicity component in a clinical trial is the repeated assessment of the treatment effect across several decision points. We will go over main components of standard classical group-sequential designs that have been used in confirmatory trials for several decades now. Section E1 will be used mainly to help us prepare for a discussion of more challenging problems that arise in group-sequential trials with multiple objectives. This will be the main topic of Section E2. We will consider the steps a clinical trial sponsor needs to take to accommodate several multiplicity components. For example, multiple endpoints and multiple doses in a group-sequential setting. Example 4, based on a prostate cancer trial with 2 endpoints. If you remember, those endpoints were progression-free survival and overall survival. This clinical trial example will be used throughout this section to help answer a popular question that I mentioned in Module A. And the question is, how do we perform multiplicity adjustments in oncology trials, or more generally in clinical trials with several endpoints that are analyzed at different time points. Section E1, Introduction to Group-sequential Trials. Let us begin with a more general discussion of the role of sequential data monitoring in confirmatory clinical trials. We know that safety and efficacy monitoring has become an integral part of late-stage clinical trials. These approaches are quite popular in phase 3 trials, but they're also employed in phase 2 trials. Interim monitoring is performed in a group-sequential manner, which means that interim looks are taken after groups of patients have completed the trial. Sequential monitoring of safety and efficacy data is driven in general by the following ethical and financial considerations. Beginning with ethical requirements, it is broadly recognized that it is critical to ensure that patients are not exposed to harmful therapies. This immediately implies that a clinical trial must be stopped as soon as the experimental treatment is found to cause serious side effects. And in addition to this, if the treatment demonstrates superior efficacy compared to the control at an interim analysis, it will be unethical to continue the trial, and the trial will be stopped at that point with a positive result. From a financial perspective, to make the optimal use of research and development dollars, early stopping due to futility would be generally desirable if a trial is unlikely to achieve its objectives. To continue and also to elaborate a bit, an option to stop a trial at an interim look is a very important option in group sequential and also in general adaptive trials. Trials may be stopped as soon as enough information is accumulated to reach a conclusion, which includes, as we said on slide 5, a positive conclusion, which means stopping due to superior efficacy, or it could be a negative conclusion, for example, stopping due to futility. Another important option is to update or revise the trial design to incorporate new information which became available at an interim analysis. This option will be more relevant in the context of adaptive designs that are aimed at, for example, selecting the best treatment dose or the best patient population based on a assessment of interim analysis results, and we will talk more about adaptive designs of this kind in Module F. In group sequential trials, only two data-driven decisions are supported. It's a decision to terminate the trial due to futility or due to superior benefit. Group sequential trials. This slide defines the general setting we're going to assume throughout this section that deals with traditional group sequential designs. We will assume, as I said, that there's only one source of multiplicity, which is induced by the repeated assessment of the treatment effect across multiple decision points. For this reason, we're going to assume that the clinical trial has a single objective, which is characterized by the fact that there's a single endpoint, single primary endpoint. The treatment effect will be evaluated in a single patient population, and there will be a single dose of the experimental treatment compared to appropriate control. We're going to assume for simplicity the case with two decision points, which means that there will be one interim analysis pre-specified, which supports an option of early stopping due to futility or due to superior treatment benefit, and an analysis of the treatment effect will be performed at the final look. I would like to point out that there are numerous important methodological issues such as computation of bias-adjusted point estimates or confidence intervals for the treatment differences in group sequential trials, as well as many other important issues that I'm not even going to mention in this section. We also need to bear in mind various logistical issues in adaptive designs related to the process of setting up data monitoring committees, enrollment management. We need to be mindful of site management issues and drug supply in adaptive designs, just to mention a few issues specific to designs with data-dependent decision rules. But our main focus in this module will be tools for multiplicity adjustment. And if you would like to learn more about group sequential trials or about general adaptive approaches to designing clinical trials, I would recommend the following 3 books. The first book, Group Sequential Methods with Applications to Clinical Trials, is probably the most popular textbook on group sequential trials. And the other 2 books, Statistical Monitoring of Clinical Trials, a Unified Approach, and Group Sequential and Confirmatory Adaptive Designs in Clinical Trials. They serve as an excellent source of information on many theoretical as well as practical considerations in group sequential trials and in general adaptive trials employed in confirmatory clinical trials. To introduce general statistical methods employed in classical group sequential trials with a single clinical objective, we will need to introduce some notation. We will assume that theta denotes the true treatment difference. For example, if the primary endpoint is continuous, then theta would correspond to the mean treatment difference between the experimental treatment and control. Under the null hypothesis of no effect, theta is equal to 0, and we're going to assume a one-sided alternative denoted by k. Under this alternative, theta is equal to theta 1, which is a positive treatment difference. Our goal here is to discuss statistical methods that help us perform multiple looks at the data to be able to test the null hypothesis of no effect in a way that the overall type 1 error rate is controlled at a one-sided alpha of 0.025. Here's a diagram that captures key points about traditional group sequential trials. We will be using similar diagrams multiple times throughout this module, so let us spend some time on familiarizing us with the general setup of this diagram. First of all, we see a time axis, or in general this would be the information axis. It ranges between 0 and the information fraction of 0, t is equal to 0, corresponds to the study start. The other extreme here is the information fraction of 1. That's where the final analysis will be performed, and an interim analysis may be performed somewhere in between. For example, it may correspond to the information fraction of t1. We're going to assume throughout this module that the information fraction that corresponds to the interim analysis is 50%, but of course the same approach can be applied to a much more general setting. And I guess I should have said at the very beginning how we define the concept of an information fraction. The information fraction t, which again ranges between 0 and 1, is based on the number of patients in fixed designs or in designs with a fixed patient follow-up, or in time to event trials it would be expressed in terms of the target number of events. From a multiplicity perspective, the most important feature of a group sequential trial is the fact that a repeated significance testing approach is applied, which means, in simple terms, that the null hypothesis H that we defined on slide 11 will be tested multiple times throughout the trial based on the accumulated evidence at each decision point. Needless to say, if the treatment effect is assessed at each decision point without a proper adjustment, for example at the full alpha level, then it's quite easy to show that the overall type 1 error rate will be inflated. To address this inflation problem, the significance levels that will be used across the decision points must be adjusted. So instead of adjusting p-values, as it is commonly done in other multiplicity settings, in this case it is the significance levels that get to adjust it, with the ultimate goal of controlling the overall type 1 error rate. And the adjusted significance levels define the so-called stopping boundaries that I'm going to define on the slide. But before we get to the discussion of stopping boundaries, I would like to quickly mention or introduce the test statistics denoted by Z1 and Z2. This is the treatment effect test statistics that will be computed at the interim analysis and at the final analysis respectively. And at the interim or final analysis, we can also compute one-sided p-values. They will be denoted by P1 and P2. For either test statistics or treatment effect p-values, we can compute appropriately adjusted stopping boundaries. The adjusted critical values for the test statistics will be denoted by U1 or U2. Here I'm using uppercase U. And a similar equivalent, actually mathematically, stopping boundary can be computed on the p-value scale. In this case, we're going to use the lowercase u. Lowercase u, 1 will denote the adjusted significance level at the interim analysis, and lowercase u2 will denote the adjusted significance level to be applied at the final analysis. So returning to the diagram with the information axis, here is the simple rule that will be applied in traditional group sequential trials to determine whether or not there is enough evidence to reject the null hypothesis of interest at the interim analysis. The decision rule is quite straightforward. If we focus on the p-value scale, then a decision to reject the null hypothesis H and stop the study early due to superior efficacy will be made if P1 is less than or equal to U1. Once again, U1 here is not alpha, it is the adjusted significance level that helps us control the type 1 error rate across the two decision points here. If this condition is not satisfied, if P1 actually exceeds U1, then a decision will be made to continue to the final analysis. This slide defines the decision rule at the final analysis. We assume here that the null hypothesis was not rejected. It was accepted at the interim analysis, and that is represented by this gray circle with the letter A. A here stands for accept, and therefore we're now interested in testing the same hypothesis of no effect at the final analysis. Of course, we will not be using the same p-value, we'll have more data, and using the additional data that will be accumulated by the time of the final analysis, we will compute another treatment effect p-value, which is denoted by P2, and that P2 will help us define a decision rule for this null hypothesis. This null hypothesis will be rejected, which indicates a significant treatment effect at the final analysis if P2 is less than or equal to U2, which is the adjusted significance level computed for the final analysis. If this condition is not satisfied, then the conclusion that will be made will be that there's not enough evidence to conclude a positive treatment effect at the final analysis. The most popular approach to carrying out repeated significance tests on accumulated data in group sequential trials relies on the so-called type 1 error rate spanning function. They're also known for simplicity as alpha spanning functions. And an alpha spanning function is a predefined function that streamlines the process of computing stopping boundaries or adjusted significance levels across the decision points in a trial. As you can see on the slide, the alpha spanning function is denoted by alpha. It's a function of t, which is the information fraction, and it's a non-decreasing function of its parameter. The alpha spanning function is equal to 0 at the beginning of the study when the information fraction is 0, and at the end of the trial when the information fraction is equal to 1, the alpha spanning function is equal to alpha. Two popular stopping boundaries, known as the O'Brien-Fleming and Pocock stopping boundaries, can be approximated using simple alpha spanning functions that you see at the bottom of the slide. It's alpha times t, that's the function that approximates reasonably well the Pocock stopping boundary, and alpha times t cubed would be a good approximation to the O'Brien-Fleming stopping boundary. I would like to point out that there are multiple other ways to approximate these popular stopping boundaries, and there are also other families of alpha spanning functions that are used in group sequential trials. The alpha spanning functions that correspond to the O'Brien-Fleming and Pocock stopping boundaries are shown in this figure. Very briefly, we see that with the Pocock approach to setting up stopping boundaries, and here I'm looking at the black line. So with the Pocock approach to defining a stopping boundary, the alpha is spent in a linear fashion. When we switch to the red curve, we're going to be looking at the O'Brien-Fleming alpha spanning function. In this case, it's clear that the type 1 error rate is spent very slowly early in a trial. To provide a comparison of those two popular spanning functions, with the Pocock alpha spanning function, the alpha is spent uniformly over the duration of the trial, or in other words, over the information interval that goes from 0 to 1, which means that the adjusted significance levels will be constant across the decision points, and this results in a fairly high probability of early stopping at an interim look. This is generally considered to be undesirable because it's a lot easier to justify early stopping if the treatment effect is very strong at an interim look. In some sense, a stopping boundary based on the Pocock alpha spanning function represents one extreme, and the other extreme is a stopping boundary derived from the O'Brien-Fleming alpha spanning function. We saw on slide 16 that very little alpha is spent early in the trial when this approach is considered, and this results in very small adjusted significance levels at early interim looks compared to the critical values used in traditional designs with a fixed sample size. This means that a trial will be terminated at an early interim look only if there is very strong evidence of treatment benefit. And also, an important consequence of this is that since there's very little alpha spent at an interim analysis, a higher significance level can be applied at the final analysis, which will obviously help improve the probability of success at the end of a trial, provided, of course, the trial was not stopped at the interim. And this is perhaps the main reason why the O'Brien-Fleming alpha spanning function is commonly used in confirmatory clinical trials. This slide shows us how we can use a predefined alpha spanning function. This could be a Pocock or O'Brien-Fleming spanning function to derive adjusted significance levels at an interim analysis as well as the final analysis. Here we're going to look at uppercase U's, which means that we're looking at a stopping boundary on the test statistic scale. And the first equation here shows that the U1, which is the adjusted critical value for the interim analysis test statistic, is simply computed under the null distribution as the probability of Z1 exceeding U1. That probability must be equal to the value of the alpha spanning function at T1, which is essentially tells us how much alpha we can spend by this interim analysis. Now that we have computed U1, the adjusted critical value for the test statistic at the interim analysis, we're now ready to proceed to the final analysis, in which case the information fraction will be set to 1, and compute U2, which is the adjusted critical value at the final analysis. This quantity is computed from the bivariate distribution, of Z1 and Z2, as you can see on the slide, and the probability that we can compute corresponds to the probability of not being able to reject the null at the interim analysis and rejecting the null hypothesis at the final. We're going to set this probability to the amount of alpha that we have left after the interim analysis, and that quantity is equal to alpha minus alpha evaluated at T1. I would like to emphasize that those calculations are performed under the null distribution of no effect. We assume that the hypothesis H is actually true because that is the condition that helps us control the overall type 1 error rate in the trial. This table gives us commonly used values of stopping boundaries on the test statistic scale or on the p-value scale. We're going to assume here that the interim analysis will be taken at the point which corresponds to a 50% information fraction. In other words, T1 is set to 0.5. Using the O'Brien-Fleming spanning function with a one-sided alpha of 0.025, we can compute uppercase U's or lowercase u's, the adjusted critical values or adjusted significance levels at the interim or at the final analysis. Let's take a look at the adjusted significance levels that are shown in the rightmost column in this table. We can see here that U1 is equal to 0.0026. It's obviously a very low adjusted significance level. As we said a few slides back, This is one of the important features of the O'Brien-Fleming spanning function. It supports an option to stop the study early and claim superior efficacy, but this happens only if there's very strong evidence of treatment benefit. And when we now switch to the final analysis, we see that the adjusted significance level at the final analysis, U2, is given by 0.024. It's actually very, very close to the significance level that would have been used in a traditional fixed sample design, which would be, of course, 0.025. Section E2, Group sequential trials with multiple objectives. We're now ready to focus on the main topic of this module, and to be able to do this, we're going to use example 4 based on a phase 3 trial in patients with prostate cancer. In this case, we're not going to assume anymore a simple group sequential design with a single objective. We're going to assume here the case of two sources of multiplicity. Those two sources of multiplicity defined on the slide are the fact that there are two decision points in this trial and there are two key endpoints that describe the efficacy profile of the experimental treatment. The multiple endpoints shown at the bottom of the slide are progression-free survival, or PFS. It will serve as the primary endpoint in this trial, and a key secondary endpoint will be given by overall survival. The second source of multiplicity is defined on slide 22. Multiplicity is of course induced by the fact that the treatment effect will be assessed repeatedly, first at the interim analysis and then also at the final analysis. We're going to assume that the decision rule to be applied at the interim analysis will only support an option to stop the trial early due to superior efficacy. To introduce the relevant notation, since we're no longer considering the case of a simple group sequential design, we need to define the null hypotheses of interest. Those null hypotheses are denoted by H1 and H2, and they correspond to the hypothesis of no effect of the experimental treatment on progression-free survival and also on overall survival, respectively. To control the overall type 1 error rate, we will need to specify a multiplicity adjustment that takes care of this first source of multiplicity, the fact that we're going to be assessing the efficacy profile based on two different clinical variables. We're going to consider several multiple testing procedures to control this alpha level locally, if you will, only with respect to this source of multiplicity. We're going to begin with a simple fixed sequence testing procedure, and then we're also going to consider a multiplicity adjustment based on the home procedure. To help define the general decision rules that will be applied in this group sequential trial with two clinical endpoints, let's take a look at this diagram. This diagram once again shows the information fraction axis. It defines the start of the study, the end of the study, and also the interim analysis at the information fraction T1. And this diagram reminds us of the fact that we no longer have a single hypothesis that needs to be tested at each one of those decision points. We now have two different hypotheses, H1 and H2, that corresponds once again to progression-free survival and overall survival. We also need to introduce families of p-values. The p-values for testing H1 at the interim and final looks will be denoted by p11 and p12, and likewise when we switch to the p-values for testing the hypothesis H2, also at the interim and at the final analysis, those will be denoted by p21 and p22. We're going to assume the same stopping boundary that we considered in section E1. We will assume that the adjusted significance levels or adjusted critical values are derived from the O'Brien-Fleming spanning function. The overall alpha level, one-sided alpha level, is set to 0.025, and we will assume that the interim analysis will be taken after 50% of the information is collected, so the information fraction here is equal to 0.5. The adjusted significance levels, as we have seen before, for this alpha spanning function are equal to 0.0026 at the interim look and to 0.024 at the final analysis. Let us now begin by answering a simple question. Let's first define the set of decision rules for the primary endpoint for PFS tests at the interim analysis and at the final analysis. Here we can directly apply the rules that we defined in section E1. Here we can pretend that there's only one clinical endpoint being evaluated, and therefore at the time of the interim analysis we are going to reject the null hypothesis H1, and we can stop the study early due to superior efficacy if the p-value for the PFS endpoint is significant at the predefined adjusted significance level of 0.0026. You can see this value at the bottom of the slide, and of course if this condition is not satisfied, this will not be the end of the game. We will simply make a decision to continue to the final analysis. At the final analysis, we're going to evaluate the amount of evidence to potentially reject the null hypothesis H1 again. The decision rule is going to be based on the p-value p12, and the significance level will be again derived from the O'Brien-Fleming alpha spanning function. The corresponding adjusted significance level is denoted by U2. It's equal to 0.024, and if the PFS p-value is less than or equal to this predefined significance level, we can claim a significant treatment effect on PFS, and if this is not the case, we're going to have to conclude a negative trial outcome. We now understand how to define decision rules in a group sequential trial with a single endpoint, and we're now ready to consider a more challenging set of questions related to how perform multiplicity adjustments based on PFS and OS tests at the same time. To be able to do this, we're going to consider a number of scenarios. The first scenario is going to assume that the PFS as well as OS effects will be evaluated at the interim analysis as well as at the final analysis. The overall type 1 error rate probability for this source of multiplicity for the analysis of several endpoints in this trial will be controlled using the fixed sequence testing procedure, which means that we're going to begin with the test for H1. That's the hypothesis that corresponds to progression-free survival. And if there's evidence of a significant effect on PFS, we're going to step down and take a look at the hypothesis H2, which means that we're going to evaluate the amount of evidence to claim a significant effect on overall survival. This fixed sequence testing strategy is depicted in this diagram. We can see here that at the interim analysis as well as at the final analysis, we're going to begin testing the hypothesis H1. And if it is rejected, we're going to step down to the hypothesis H2. The question we're going to focus on right now is the following. Assuming that the study will be stopped at the interim analysis due to a superior effect of the experimental treatment on progression-free survival, which means that the hypothesis H1 is rejected, what will be the appropriate adjusted significance level that can be applied to the hypothesis H2? That's the hypothesis of no effect for overall survival. I think it will be instructive to begin with a naive approach, and then I'm going to show you why this approach fails. And after that, I think we will be ready now to consider the appropriate approach that actually controls the overall alpha with respect to 2 decision points and 2 endpoints in this clinical trial. The naive argument goes like this. We're going to assess the treatment effect on overall survival only once. This is going to happen only after a significant PFS effect is established, and therefore we can spend all of the alpha on testing the hypothesis H2, which means that, as you can see on the slide, the p-value p21 will have to be compared to the full alpha level, and we're going to reject the hypothesis H2, and we're going to claim a significant treatment effect on overall survival if p21 is less than or equal to alpha. Of course, this set of rules that we define on slide 30 assume a positive effect on progression-free survival at the interim. If this is not the case, and if the PFS null hypothesis is actually accepted, which is shown on the slide by a gray circle with the letter A of course stands for accepted, we will proceed to the final analysis. We know how to test the treatment effect on progression-free survival. We're going to have to use the appropriate adjusted significance level. What about the overall survival test? Using the logic that I presented on slide 30, it would seem natural to carry out that test at the full alpha level, which means, as shown on the slide, that the p-value p22 will be compared to the full alpha level, and a significant treatment effect on overall survival can be claimed if p22 is less than or equal to alpha. It turns out, and this was actually anticipated, that the naive approach that we presented on slides 30 and 31 does not protect the overall alpha level across the two endpoints. What we need to realize here is that even though the null hypothesis H2 will be tested only once in an actual trial, there's always a possibility that it could be tested either at the interim analysis or at the final analysis. And this possibility actually induces multiplicity, which is not accounted for if we simply test the hypothesis H2 at the full alpha level. The solution to this problem is to apply either a common alpha spanning function or potentially an endpoint specific alpha spanning function to protect the type 1 error rate. What we said a few minutes ago is that we pre-specified the O'Brien-Fleming alpha spanning function for PFS due to the possibility that the PFS test could be carried out at the interim or at the final, and the same logic is actually going to have to be applied to testing the overall survival effect. We will need to pre-specify an alpha spanning function for the overall survival test. The correct strategy for testing the hypothesis H2, or in other words, for evaluating the overall survival effect at the interim analysis, is presented on this slide. The decision rule for the secondary endpoint goes like this. Instead of spending all of the alpha on the overall survival test at the interim analysis, we can only spend the amount of alpha which is available to us at this point. If we assume that the same or alpha spanning function is applied, if we assume a common alpha spanning function for the PFS and overall survival tests, then the hypothesis H2 will be rejected if p21 is less than or equal to u1, which is equal to 0.0026. And a decision rule for the secondary endpoint, overall survival, will be defined using exactly the same approach at the final analysis. Assuming once again a common alpha spanning function for the two endpoints, the same adjusted significance level will be applied for testing the null hypothesis H2, provided we can claim significance for progression-free survival at the final analysis. As you can see on the slide, we can conclude a significant treatment effect on overall survival if its p-value, p22, is less than or equal to u2. And this adjusted significance level is equal, in this particular case, to 0.024. To help understand why we need to predefine an alpha spanning function for the secondary endpoint for overall survival, it will be very helpful to examine the following scenario. We're going to refer to it as scenario 2, and the difference between scenario 1 that we have just considered and scenario 2 is that progression-free survival test will be carried out at the interim analysis as well as the final analysis, But with the overall survival test, we're going to only look at the effect on overall survival once during the study. We're going to carry out the test only at the final analysis. We're going to apply the same multiplicity adjustment based on the fixed sequence testing procedure to control for potential alpha inflation due to the analysis of multiple endpoints in this trial. Assuming scenario 2, we will very briefly define the decision rules for the primary endpoint. For PFS, they're actually going to be exactly the same as in scenario 1, because the difference between scenarios 1 and 2 was related to the way overall survival effect will be addressed. When we look at progression-free survival tests, we can claim a significant effect on progression-free survival and stop the study early due to superior efficacy if the p-value for the progression-free survival test is less than or equal to alpha 1. And moving on to the final analysis, a significant effect on progression-free survival will be claimed if its p-value at the final analysis, denoted by p12, is less than or equal to u2. If we now assume that a significant effect on progression-free survival is established at the final analysis, we're of course going to wonder what we should do to assess the effect of this experimental treatment on overall survival. We're going to step down to the hypothesis H2 as shown on the slide, and which significance level should we use for testing the overall survival effect. In this case, since there's only one possibility to assess the treatment effect on overall survival, remember we emphasized before when I introduced scenario 2 that the overall survival test can be carried out only once during this trial. It can be carried out only at the very end of the trial. Because of that, there's no alpha spending associated with the overall survival test. And because of that, we can spend all of the alpha at the final analysis, which means that we can claim a significant treatment effect on overall survival if p22 is less than or equal to alpha. That is a fundamental difference between scenarios 1 and 2. Once again, in scenario 2, there were two possibilities for us to examine the treatment effect on overall survival. This could have been done at the interim analysis or at the final, and in scenario 2, we are under no circumstances we can assess the overall survival effect at the interim analysis. For that reason, we can apply the full alpha level to the H2 test at the final analysis. The next scenario we're going to consider in this section is the scenario that assumes the same general setup as in scenario 2, which means that both PFS and OS will be tested at the interim analysis as well as the final analysis. And the positive trial outcome at the interim or final analysis will be concluded if there's a significant treatment effect on either overall survival or PFS, which means that we can no longer apply the fixed sequence testing strategy. It would be more appropriate for us to use an alpha splitting strategy of some kind. And since, as we've said so many times before, the Bonferroni adjustment is quite conservative, it would make much more sense to consider a multiplicity adjustment based on the Holm procedure. So at the interim analysis or at the final analysis, we're going to assess the amount of evidence to support a decision to reject H1 or H2 based on the Holm procedure, and we're going to split the alpha, initially at least, between the PFS and OS tests unequally. We're going to assign a larger weight, specifically W1 will be set to 0.8, which means that initially 80% of the alpha will be assigned to the PFS null hypothesis, and the remaining weight will be assigned to the overall survival null hypothesis. Of course, due to the nature of the Holm procedure, due to the option of alpha propagation, those tests will be updated when we begin testing the two null hypotheses. This slide 40, reminds us once again of the fact that the Holm procedure is a stepwise procedure that takes full advantage of alpha propagation, and the hypothesis weights W1 and W2 that I mentioned on slide 39 are only initial hypothesis weights that can be updated if we can reject one of the hypotheses. The cyclical procedure for performing this stepwise procedure is defined at the bottom of the slide. It involves 3 different tests. We can begin with the PFS test, and we can reject the corresponding hypothesis of no effect. It's the hypothesis H1 if its p-value is less than or equal to alpha times the initial hypothesis weight, which is W1. If we can reject the null hypothesis, or if it's even not rejected, we can proceed to step 2, which is going to focus on the overall survival test. Here, the significance level that will be applied to the overall survival test is going to depend, of course, on whether or not we were able to reject the PFS hypothesis in step 1. This was indeed the case. If H1 was rejected, then all of the weight that was assigned to the PFS hypothesis would be carried over to the overall survival hypothesis, and therefore we can reject H2 if P2 is less than or equal to alpha. However, if H1 is not rejected, there is still an option for us to claim a significant effect on overall survival, and this will happen if P2 is less than or equal to alpha times W2. That is the initial weight of the overall survival hypothesis. And there's also an option to come back to the PFS test. This could be accomplished in step 3, and this would be a relevant test if we could not reject H1 in the first step, but in the second step of this testing algorithm, the overall survival hypothesis was rejected. In this case, we're going to go back to the hypothesis H1, and we can reject it if the corresponding p-value P1 is less than or equal to alpha. This is again one of the important features of alpha propagation. Every time we can reject a null hypothesis, then the significance level used in that test is going to be carried over to the other hypothesis, provided of course it was not rejected. This multiple testing strategy, based on the Holm procedure, will be applied at each decision point, first at the interim analysis and then at the final analysis. We can see here are 2 sets of hypotheses that are connected by arrows, and those arrows visualize the fact that we can propagate the alpha from the PFS test to the OS test and back, and this once again can happen at the interim or at the final analysis. When we have to deal with more complex, more flexible testing algorithms that involve alpha propagation, the computation of stopping boundaries gets a bit more complicated, but it's still a fairly straightforward process. This table defines the significance levels, I should say the adjusted significance levels, that should be used at the interim analysis and at the final analysis for the PFS test. That is for testing the null hypothesis H1. We assume here once again the O'Brien-Fleming spanning function with the 50% information fraction. The first row in this table corresponds to the adjusted significance levels that will be applied at the interim analysis, and the second row defines the adjusted significance levels for the final analysis. Now let's take a look at the columns in this table. The column under the heading type 1 error rate is equal to alpha defines the adjusted significance levels that will correspond to the case when all of the weight will be set to 1. And the second column under the heading type 1 error rate is equal to alpha times W1 will correspond to the adjusted significance levels corresponding to the case when we only begin testing the null hypothesis H1. We're not coming back to it in stage 3. I know it may sound a little bit confusing, so let me walk you through this process very carefully, one slide at a time. In addition, we will need to define a stopping boundary for the overall survival test. and will be based on exactly the same principles. In fact, the only difference between this table and the table that I gave you on slide 42 is that the adjusted significance levels in the rightmost column in this table are slightly different. They are smaller than the adjusted significance level shown in the table on slide 42, simply because the weight initially allocated to the overall survival test is smaller. Instead of using W1, which is equal to 0.8, here we're going to assume that W2 is equal to 0.2, which results in smaller adjusted significance levels. So where exactly did those adjusted significance levels come from? If we begin with the process of spending the alpha for the progression-free survival test, that is for the hypothesis H1, there are two alpha spending functions displayed in this figure. The alpha spending function, represented by the black curve, is simply the standard O'Brien-Fleming alpha spending function, which corresponds to the alpha of 0.025, and this alpha spending function will be used to derive the adjusted significance levels that will be applied if the Holm procedure tests the hypothesis H1 at the full alpha level. This will happen, for example, in step 3 of the Holm testing algorithm. However, in step 1 of the same algorithm, we know that the hypothesis H1 will be tested at a reduced significance level, which is given by alpha times W1, and to be able to compute the corresponding adjusted significance levels that will be used in the group-sequential setting, we will need to apply the red curve. That's the alpha spending function that corresponds to the total error rate of alpha times W1. Of course, as you can see in this figure, the adjusted significance levels derived from the red curve will be slightly smaller than the adjusted significance levels based on the black curve. And that's exactly the information that we saw summarized on slide 42. The same logic will be applied to compute the adjusted significance levels for the hypothesis H2, that is for assessing the overall survival effect at the interim or at the final analysis. Just like in the figure I presented on slide 44, here we see two curves. The black curve defines the standard alpha spending function that will be applied when the Holm procedure carries out the test for H2 at the full alpha level. This will happen in step 2, and the red curve will be applied when the hypothesis H2 is tested by the Holm procedure at a reduced level. Specifically, the level is given to alpha times W2. We saw the corresponding adjusted significance levels summarized in the table given on slide 43. We have now defined all of the key components of the decision rules that will be applied at the interim analysis for progression-free survival and overall survival under scenario 3. So let's now take a look at the specifics. We're going to begin with the interim analysis, of course. Let us first take a look at the decision rules for testing the progression-free survival effect. As you can see on the slide, the p-value for the PFS test, p11, will be compared to the adjusted significance level, which is derived from the appropriate alpha spending function, assuming that the Holm procedure tests this null hypothesis at the reduced level given by alpha times W1. This adjusted significance level, as you can see at the bottom of the slide, is equal to 0.0017, and therefore a significant treatment effect on progression-free survival can be claimed here if the PFS p-value is less than or equal to this particular quantity. Now we're going to assume that the hypothesis H1 was rejected at the interim analysis. Then what will be the significance level that can be applied to the hypothesis H2? This slide helps answer this question. We can refer to this as step 2a. This will be the set of decision rules that will be applied to the overall survival test, provided we were able to reject the null hypothesis H1 for progression-free survival. In this case, the p-value p21 will be compared to the adjusted significance level, which corresponds to the case where the Holm procedure tests H2 at the full alpha. This adjusted significance level is shown at the bottom of the slide. This is actually the standard adjusted significance level, which is derived from the O'Brien-Fleming alpha spanning function. It is 0.0026, and we can now claim a significant effect on overall survival if its p-value is less than or equal to this adjusted significance level. However, there is no guarantee that the PFS test will be rejected at the interim analysis. It's possible that H1 will not be rejected, in which case this will be reflected in the adjusted significance level that will be applied to the overall survival test. This is exactly the scenario which is considered on slide 48, and we're going to refer to this as step 2b to differentiate this from step 2a, where we assumed that the null hypothesis H1 was rejected. Here, we cannot use the full alpha level to derive the adjusted significance level, and as you can see on the slide, the adjusted significance level here is computed under the assumption that the Holm procedure in this step tests the hypothesis H2 at the reduced level given by alpha times W2. This results, of course, in a much more stringent set of conditions under which the null hypothesis H2 can be rejected. We can see at the bottom of the slide that the corresponding adjusted significance level is only 0.001. The last case we need to consider here corresponds to step 3 of the Holm procedure. In this case, we need to assume that H1 was not rejected in step 1, that is the first time around. In addition to this, we need to make an assumption that the hypothesis H2 was rejected, and that enables us to come back to the hypothesis H2, and all we need to do now is to figure out which adjusted significance level can be used in this test. The answer to this question is shown on the slide. We can see here that we need to compute the adjusted significance level under the assumption that the Holm procedure tests this hypothesis H1 at the full alpha level, which results in the adjusted significance level of 0.0026. So to summarize, we come to a conclusion that there is a significant treatment effect on progression-free survival if the corresponding p-value is less than or equal to 0.0026. Before we proceed to the final analysis, I would like to mention an important set of rules that are applied in group sequential trials with multiple objectives. First of all, a trial may potentially continue to the next decision point. For example, it may continue from the interim analysis to the final analysis, even if superior efficacy is established at the interim analysis. If the trial sponsor decides to go down this path, they will need to apply the following set of consistency restrictions. If a hypothesis is rejected at the current decision point, for example, at the interim analysis, it will not actually need to be retested at the final analysis. It will be automatically rejected at all subsequent decision points without testing. Keeping this consistency restriction in mind, it will be now sufficient for us to consider the decision rules at the final analysis under the assumption that we were unable to reject either hypothesis at the interim analysis. Those accepted hypotheses, as we have done before, are represented here by the grayed out circles with the letters A. So what will be the significance levels that can be now applied to the hypothesis H1 and H2 at the final analysis? We will need to apply the same approach that we applied at the interim analysis. The only difference here is that the adjusted significance levels will be computed from the alpha that is available to us at the interim analysis. And therefore, instead of using lowercase u1, we will now start using lowercase u2. These are the adjusted significance levels that were computed for the final analysis in the tables that we looked at a few slides back. So we're going to once again look at the individual steps of the testing algorithm used by the Holm procedure. In step 1, we're going to reject the null hypothesis H1 that corresponds to the progression-free survival test if the corresponding p-value is less than or equal to the adjusted significance level derived from the significance level that would have been used by the Holm procedure in step 1. And that significance level is alpha times W1. As we have done before, we're going to now take a look at two different cases that correspond to step 2. We can assume that the hypothesis H1 was rejected in step 1 or it was not rejected in step 1. In this particular case, we're going to make a more optimistic assumption. We're going to assume that the hypothesis H1 was actually rejected in step 1, and therefore that enables us to use all of the alpha in the computation of the adjusted significance level for the overall survival p-value. This resulting adjusted significance level is shown in the slide, and the numerical value of this adjusted significance level is 0.024. That is the value of the threshold that will be used in assessing the amount of evidence to reject the hypothesis H2 based on overall survival at the final analysis. Of course, we should not forget about the case when the hypothesis H1 was not rejected in step 1. In this case that we can refer to as step 2b is presented on the slide. In this case, we can still, of course, test the hypothesis H2 for overall survival, but the price we'll have to pay for failing to reject H1 in the first step is that when we compute the adjusted significance level, it will be computed based on the reduced significance level that is used in the Holm testing algorithm. The resulting threshold is equal to 0.0049, and that is the threshold that will be used in the decision rule for rejecting H2. The last case we need to consider here is represented by step 3, which corresponds to the following set of outcomes. We assume that the first time around in step 1, the hypothesis H1 was not rejected. There was not enough evidence to claim a significant effect on progression-free survival at the final analysis. We then proceeded to test the overall survival effect and were able to reject the hypothesis H2. This enables us to come back and retest the progression-free survival hypothesis, or H1, and this will be done at a higher significance level. To be able to compute the corresponding adjusted significance level, we begin with the full F level. That is the significance level that would have been used by the regular Holm procedure in step 3. And then, using this as a starting point, we apply the O'Brien-Fleming alpha spanning function, and that gives us the appropriately adjusted significance level. This significance level is shown at the bottom of the slide. We have seen it multiple times before. That is the standard significance level derived from the O'Brien-Fleming alpha spanning function, it's 0.024, and that is the level that will be used in step 3 to decide whether or not we have enough evidence to reject the hypothesis H1. I'm going to spend just a couple of minutes on scenario number 4. We're not actually going to go into the actual numeric example. Here I'm going to use this scenario mostly to remind you of the fact that we don't have to specify exactly the same alpha spanning function for all of the multiple endpoints considered in a group sequential clinical trial. It is quite possible to define endpoint specific alpha spanning function. For example, as you can see on the slide, we may decide to use the standard O'Brien-Fleming spanning function for the progression-free survival test, but another, for example, the Pocock spanning function for the overall survival test. An advantage of this approach is that it helps us improve the probability of success for overall survival. We know that when we compare data maturity for those two endpoints, PFS and OS, data tends to be less mature for the overall survival test, and therefore using a more generous, if you will, alpha spanning function, such as the Pocock spanning function, helps us improve power of the overall survival test at the interim analysis. Assuming that the same multiplicity adjustment based on the Holm procedure is going to be used, the weight will be set to 0.8 for progression-free survival and to 0.2 for overall survival. The decision rules will be derived in exactly the same fashion as in scenario 3. The only difference here is that we'll need to compute the adjusted significance levels for the overall survival test differently, because here we assume a different spanning function. To remind you once again, we decided to test the effect of the experimental treatment on overall survival using the Pocock alpha spanning function, and that gives us a slightly different set of stopping boundaries based on the full alpha level or based on the reduced significance level, which is given by alpha times W2. You can compare the resulting adjusted significance levels, and you can see here that this is a more liberal approach at the interim analysis. There's going to be, as I promised on slide 55, a higher probability of claiming a significant effect on overall survival due to the fact that we're going to spend more alpha by the time of the interim analysis using the Pocock alpha spanning function. The last topic we're going to consider in this module is software implementation. As in modules C and D, I would like to very quickly mention software implementation options for group sequential designs in SAS as well as in R. In SAS, there's a built-in procedure called seq design procedure. It obviously stands for sequential design. It can be used to compute the adjusted significance levels based on popular alpha spanning functions. I would like to mention this quickly as an option. I'm not going to actually go into explaining how to use this procedure. As an alternative to using SAS, we can spend a bit more time on discussing how similar calculations can be performed using open source software. Specifically, there's a popular package called the GS Design package available in R that can be used to perform similar calculations. It supports calculations of adjusted significance levels across multiple decision points based on commonly used alpha spanning functions. You can learn more about this particular package by visiting the package's web page on the CRAN website. You can see the link on the slide. To support calculations for the adjusted significance levels based on the O'Brien-Fleming spanning function, we will need to run the following code. You can see here that it takes only one line of code, and all we have to do is to call the GS design function and simply supply the appropriate arguments. Assuming that we have a clinical trial with two decision points, which means a single interim analysis at the 50% information fraction, and assuming that the overall type 1 error rate is set to 0.025, the following function call will need to be made here. First of all, the first argument of this function, k is equal to 2, specifies the number of decision points. It's once again the interim analysis plus the final analysis. Second argument, timing, defines the information fractions at the interim analysis and at the final analysis. Those values are given by 0.5 and 1. The third argument, test type, is equal to 1, because in this case we have a single stopping boundary, and this is the stopping boundary that supports decisions to claim superior efficacy at the interim analysis or at the final analysis. The fourth argument of this function specifies the O'Brien-Fleming alpha spanning function. The overall alpha is given by 0.025, and in fact the last argument in this function, which is the beta or type 2 error rate, is not required for this particular calculation. It becomes an optional argument and it could be potentially omitted. When we run this function, the output of the function is shown at the bottom of this slide, and the column that we need to pay most attention to is the column with the heading nominal p. There are two values shown in this column. The value which corresponds to the analysis 1, that's the interim analysis, is 0.0026. This is the adjusted significance level produced by the O'Brien-Fleming spanning function for the interim analysis, and for the final analysis the adjusted significance level, as you can see on the slide, is given by 0.024. If we are interested in performing similar calculations based on the Pocock spanning function, the same code can be run with one very simple modification. We can see here that the function call here is exactly the same as on slide 60. The only difference is that we have replaced the alpha spanning function. Instead of OF, which stands for O'Brien-Fleming, we have now used the value of Pocock. We can now run this function and it gives us the adjusted significance levels at the interim analysis as well as the final analysis. With the Pocock spanning function, these adjusted significance levels are always the same, and it's equal to 0.0147 at the interim as well as the final analysis. The last calculation I'm going to present in this module deals with the computation of adjusted significance levels for group-sequential designs where multiplicity adjustments based on procedures such as the Holm procedure that enable alpha propagation can be used. With procedures such as the Holm procedure, we may need to predefine hypothesis weights. We assume here scenario number 3, and suppose that the predefined hypothesis weight for the PFS test is equal to 0.8, that we can easily modify the function call presented on slide 60 to compute the corresponding adjusted significance level. All we need to do here is to change the value of the alpha parameter. Instead of applying the full alpha level, we're going to apply a reduced significance level, which is equal to alpha times W1. And after we call this function, we're going to compute the adjusted significance levels that needs to be used in step 1 of the Holm procedure. These adjusted significance levels at the interim analysis as well as the final analysis are displayed at the bottom of the slide. We can see here that they are equal to 0.0017 at the interim analysis and to 0.0193 at the final analysis. This is the kind of code that I ran to populate the tables I used in scenarios 3 and 4.